Today we will continue our discussion about molecular biology, and today's topic is DNA and RNA chemical structure. So we have two types of nucleic acids. It's depending on its sugar. It will be called deoxyribonucleic acid or ribonucleic acid. Both of it have ribose sugar. One ribose misses its oxygen, and that's why it's called deoxyribose sugar. And here you have ribose sugar, which contains its oxygen in place, right? And what else? They differentiate by having one of it, like DNA has two strands and RNA have only one strand, and by its nitrogen bases, where they differentiate by thymine and uracil bases because DNA contains thymine and RNA contains uracil base instead. Here we will take a closer look to our DNA, and this backbone of DNA actually contains nucleotides. Each nucleotide contains three parts of it, which is phosphate part, sugar part, and base. And we have four types of bases. It is a nitrogen bases, adenine, thymine, guanine, and cytosine. When we form backbone, it actually will connect with each other with phosphodiesterate bond, which is really strong and really hard to disconnect. Only when damage or mutation occurs it's possible. And we have more weak bonds, which is hydrogen bonds between nitrogen bases, and we actually always connect adenine with thymine and guanine only with cytosine. And when we connect adenine with thymine, we have two hydrogen bonds, and when we connect guanine with cytosine, we have three hydrogen bonds. And when we form two hydrogen bonds is actually obviously weaker than when we have three hydrogen bonds. And if DNA is if any part of DNA contains more guanine and cytosine in it, it will actually be harder to melt, and it will take more time to do so in order to make transcription, in order to make some protein or make DNA replication to make new strand of DNA. Here you can see that we make RNA first, and then from this RNA we make protein. Here we translate it um, to nucleic acid language, and then we make proteins out of it. Here you can see DNA sequence, which contains like nucleotides in it, and it then makes another nucleic acid from it, RNA nucleic acid, in order to make protein afterwards. And you can see three nucleotides called one amino acid, and if you bind this amino acid, you make polypeptide chain, and after it will proliferate to mature protein. Nitrogen bases divided to purines and pyrimidines in order to memorize which ones is purines, which ones is pyrimidines. We have mnemonic in first aid. Pure as gold stand for purines for adenine and guanine, which contains two rings in it, like here you can see. And pyrimidine stands for cut the pie, like uh, pyrimidines is cytosine, uracil, and thymine, which contains only one ring. We will talk about purine and pyrimidine de novo synthesis later, but here I just want to tell that purine contains three amino acids. In order to make purine, you have to have three amino acids, which is glycine, aspartate, and glutamine. And for this as well, we have mnemonic, which is Cas pure until they gag. So after they gag, they're not pure anymore. So GAG stands for glycine, aspartate, and glutamine. Here you can see double-stranded DNA structure, and it is a backbone, and two backbone goes anti-parallel to each other. One goes from 5 prime to 3 prime, other goes from 3 prime to 5 prime, as you can see, and it binds by phosphodiesterase bonds from 3 prime to 5 prime, from 3 prime to 5 prime, and here you can see from 5 prime to 3 prime, 5 prime to 3 prime, and it bases the binds which is each other in the middle with a weak basis in order to be able to melt afterwards. Uh, Between thymine and adenine, we have two hydrogen bonds. Between guanine and cytosine, three hydrogen bonds. So the amination reaction occurs when there's DNA metabolism or when there's DNA damage in order to repair. So we have two types of DNA damages. It is from etiology. It can be endogenous or exogenous. When it's endogenous, it actually occurs by itself inside our body, and we can just regulate it by mismatch repair mechanism when DNA polymerase just goes back and repairs its mistake by itself. And we have mechanisms by chemical reaction, like first we deaminate it, and when we deaminate certain base, we create something new. Like from cytosine, we create uracil because it has similar structure, and after that we just um, make excision. So uh, after we convert it to uracil, we just make excision of uracil, and our backbone stays in its place. So we just uh, repair it by DNA polymerase afterwards. And we also make it with adenine. We just deaminate it first, make hypoxanthine, and then excite it, and create new proper base instead. And here you have depurination, which is same mechanism, but here you just cut off your purine, and backbone stays in its place, and then you just um, repair it by creating new base in its place. Here you have exogenous reasons. For example, under UV radiation, you make pyrimidine dimers, which creates formic cytobutyric ring, and it can make helix distortion, and it will create melanomas, skin melanomas. And also we have basic excision repair mechanisms when there's a methyl group or ethyl group will be added. It occurs when organism is exposed to certain carcinogens, for example, cigarette smoke, and it will actually creates disruption of hydrogen bonds between nucleotide, and it will distort helix and cause frame shift mutation, which leads to, for example, lung cancer when we talk about smoke. I forgot to mention DNA brick. So DNA bricks are made of nucleotides, and here you can see nucleotide is when we combine base, sugar, and phosphate together. Without phosphate, it calls nucleoside. and with phosphate it calls nucleotide. And these other phosphate groups, we can have three phosphate groups. This first one we contain to just create phosphodiester bonds, right? In order to create backbone of our nucleic acid, and this too just creates energy to this bond to be created, right? Without any energy, we cannot create this bond. To memorize, we have mnemonic from first state. Nucleoside contains sugar with S in it. Nucleotide contains phosphate with T in it.